The brain is an organ so complex and fascinating that it's often referred to as the most complicated object in the universe. Composed of approximately 86 billion neurons, the brain is a masterful piece of biological engineering. It's divided into different lobes: the frontal lobe, the parietal lobes, the temporal lobes, and the occipital lobes. Each region specializes in processing certain kinds of information while simultaneously working together with other regions in the brain for perception, thought, and emotions. To truly understand the brain, though, we need to zoom in, way in, to the most fundamental building blocks of thought, perception, and information processing: the neurons. Imagine a neuron, the fundamental building block of our nervous system, as a living, breathing tree. It's not just a static structure, but a dynamic entity constantly interacting with its environment. The roots of this tree, akin to the dendrites of our neuron, mark the beginning of a fascinating journey of information processing in the brain. Picture the roots spreading out, reaching into the soil, absorbing life-giving water and nutrients. Similarly, the dendrites of a neuron extend their reach, eager to receive information from their neighboring neurons. This information exchange happens through neurotransmitters, the chemical couriers of our nervous system. They bridge the gap, or synapse, between neurons, much like a message in a bottle crossing a river. When a neuron is sparked into action, it releases these neurotransmitters into the synapse. They then find their way to specific receptors on the dendrites of the next neuron, like a key finding its way into a lock. Each neurotransmitter has a corresponding receptor it can bind to, ensuring the right message gets to the right place. Once the neurotransmitter finds their home in the receptors, they set off a chain of chemical reactions that can either stimulate or inhibit the neuron's activity. It's like a domino effect, with each reaction leading to the next. When the neuron is stimulated, these chemical changes travel to the cell body, the trunk, or the hub of our info-processing neuron. This is where the neuron's nucleus resides, along with other vital components. Here, the chemical changes from all the dendrites are summed or integrated, much like a river gathering strength from its tributaries, growing larger and more powerful with each addition, before being sent on to the axon. The axon, our tree's branches, then carries this information, often across vast distances, to the axon terminal. And finally, we arrive at the axon terminal, the leaves of our tree. This is where the information is passed on to the next neuron, continuing the cycle of communication in this intricate network of neurons. When we group these neurons, we form a structure known as a nerve. These nerves can span considerable distances. For instance, the sciatic nerve, which you may have heard of, extends from the lower back all the way down to the feet. I've previously mentioned that the information that travels along the dendrites through the axon and onto the axon terminal is facilitated by chemical changes. But what does this mean? Let's delve into it. The dendrites, which are part of the neuron, receive incoming signals. If the stimulation is potent enough, this signal is transmitted along the entire length of the axon in a process known as an action potential. This is when the neuron is said to fire. The transmission of neuronal signals rely entirely on the movement of ions, or charged particles. These are the chemical changes I refer to. Various ions, including sodium, potassium, and chloride, move between the inside and the outside of the cell. The role of these ions is not limited to when a neuron fires. They are essential during the neuron's resting state. Let's take a closer look at the sodium and potassium ions, both of which carry a positive charge. In the resting state, when a neuron is not transmitting any signals, the distribution of these ions is quite specific. Sodium ions are found in greater concentration outside the cell, while potassium ions are more concentrated inside the cell. This separation of ions happens at the cell membrane, establishing a chemical gradient across it. Given that ions are charged particles, their charge is a crucial factor in their distribution across the membrane. At rest, there are more positively charged ions outside the cell compared to inside. This difference in charge across the membrane, known as the electrical gradient, combined with the chemical gradient, forms what we call the electrochemical gradient. The disparity in total charge inside and outside of the cell is referred to as the membrane potential. In a resting state, when no signals are being transmitted, the neuronal membrane has a resting potential of approximately minus 70 millivolts. This indicates that inside of the cell is approximately 70 millivolts less positive than the outside. This is similar to the concept of electrical potential in this 9-volt battery. The battery has a positive end and a negative end, and the difference in the electric charge between these two ends creates an electrical potential, or voltage. In the case of the battery, it's a 9-volt electric potential. This potential is what drives the flow of electrical current when the battery is connected in a circuit. This resting potential is maintained by ion pumps and channels that regulate movement of positively charged ions, like sodium and potassium, and negatively charged ions, like chloride, across the neuron's membrane. The chemical and electrical gradients we've discussed are key to establishing this potential. Although the cell's interior carries a net negative charge and the exterior a net positive charge, these charges align at the membrane. Interestingly, the overall solution on either side remains electrically neutral. The resting membrane potential signifies the point of electrochemical equilibrium for the cell. This is the state where the concentration gradient and electrogradient for each ion balance each other out. Ions don't have free passage across the membrane. Their movement requires a protein embedded in the membrane. Most ions traverse the membrane through a structure known as an ion channel. Ions move through these channels by passive diffusion along their concentration gradient. Some ion channels are perpetually open, while others need a specific signal to open or close. For instance, voltage-gated channels open when the membrane potential hits a certain threshold. Ligand-gated ion channels, on the other hand, open when bound by a specific molecule. And mechanically gated ion channels respond to physical forces, such as changes in length or pressure. As a quick aside, these mechanically gated ion channels are particularly important for hearing and touch and balance. For example, when you touch something, the pressure from that touch deforms the skin and applies a force to the cutaneous receptors, which are in fact neurons. This force can cause mechanically gated ion channels to open, leading to an influx of ions and the generation of an electrical signal. This signal is then transmitted to the brain, where it is interpreted as the sensation of touch. In the cochlea, the part of the inner ear involved in hearing, sound waves cause the fluid-filled chambers to vibrate, and these vibrations deflect the hairs known as Stereocilia on the hair cells, which are connected to mechanically gated ion channels. When the Stereocilia bend, these channels open, allowing ions to flow into the hair cell. This ion flow generates an electrical signal that is sent to the brain via the auditory nerve, where it is interpreted as sound. For all neurons, most ion channels are selectively permeable, meaning they only permit one or a small group of ions to pass through. Voltage-gated ion channels, for example, typically allow only a single type of ion to cross the membrane when they open. This necessitates separate channels for each ion, such as voltage-gated sodium channels and voltage-gated potassium channels. As ions traverse a channel and cross from one side of the cell membrane to the other, they cause the membrane potential of the cell to deviate from its resting potential. If the resulting change in membrane potential is minor, we refer to this as a graded potential. Graded potentials are like the whispers of the neuron. They can fluctuate in intensity, be positive or negative, and are fleeting, and usually do not result from the opening of voltage-gated ion channels. When ion channels open and a graded potential takes place, the neuron swiftly works to reset its membrane potential to resting values. This reset is primarily achieved by the sodium-potassium pump, a diligent worker that uses energy from ATP hydrolysis to actively transport ions across the membrane against their concentration gradient. In essence, sodium is shuttled to the outside of the cell, where it's more concentrated, and potassium is ushered back into the cell, where it's more abundant. A single cycle of this pump moves 3 sodium ions outside the cell and brings 2 potassium ions inside. This unequal charge transfer aids in maintaining the charge separation across the membrane and also the ionic concentrations at rest, thus restoring the chemical and electrical gradients to their resting levels, such as minus 70 millivolts. The brain as a whole represents about 20% of the body's energy consumption, which given its size is a huge amount. Preserving this ionic balance in neurons is so crucial that it can account for a significant amount of that energy. Only when the resting membrane potential and ion distributions are kept at precise levels is the neuron primed and ready to fire an action potential. When the external stimulation received through the dendrites is potent enough to elevate the membrane potential in the neuron from minus 70 millivolts to the threshold voltage of minus 55 millivolts or higher, this triggers an action potential at the axon hillock, which then travels down the axon. Voltage-gated sodium channels are the driver, and they have 3 states: open, closed, and inactivated. At rest, the sodium channel is closed. Once the cell membrane reaches the threshold voltage, the channel transitions to an open state, and sodium floods into the cell due to the electrochemical gradient. As positive sodium ions enter the cell, the membrane potential shifts from negative towards positive, reaching 0 millivolts. This shift is known as depolarization. Eventually, the voltage gradient crosses 0 and ascends to a positive 30 millivolts. This is referred to as an overshoot. As the membrane potential turns positive, the sodium channel in activation gate closes, halting the influx of sodium ions into the cell. The change in membrane potential also opens the voltage-gated potassium channels, although they open and close more sluggishly. Due to the sluggish nature of potassium channels, there's a brief moment when the membrane potential dips below the resting potential, becoming hyperpolarized. During this hyperpolarization phase, the potassium channels eventually close. All the while, the sodium-potassium pump tirelessly continues its work. It restores the chemical gradients by repositioning the sodium and potassium ions. Simultaneously, it reestablishes the potential gradient by expelling more sodium ions than it takes in potassium ions. This diligent activity brings the membrane potential back to its resting state. During the repolarization phase, the inactivated sodium channels become unresponsive to any stimulus. This period is known as the absolute refractory period, and it's when a neuron is incapable of firing another action potential, regardless of the strength of the stimulus. The absolute refractory period serves as a safeguard, preventing action potentials from occurring too rapidly or moving in the wrong direction along the axon. During hyperpolarization, the sodium channels are closed and the inactivation gate a little plug that ensures no sodium can pass through, opens. There's no change in sodium flow, but they are now primed to open again. This phase is known as the relative refractory period. Although the sodium channels could open, it would require a stronger than a usual stimulus to reach the threshold, as the cell is hyperpolarized due to the ongoing outflow of potassium. The peak voltage of an action potential for specific neuron, that is, the maximum voltage reached during an action potential, remains constant. An action potential doesn't amplify with a stronger stimulus. It's an all or nothing event. It either occurs or it doesn't. What can vary, however, is the frequency of the action potential. A neuron might fire more rapidly in response to, say, intense pain, and less frequently in response to a gentle breeze. Certain axons transmit action potentials faster than others. One factor that boosts conduction speed is the presence of myelin sheaths around axons. Myelin accelerates transmission through a process known as saltatory conduction, where the action potential signal seems to leap along the myelin-covered part of the axon. In diseases like multiple sclerosis, or MS, it is these myelin sheaths that the immune system mistakenly attacks, leading to demyelination. When myelin is damaged or destroyed, the communication between neurons can be disrupted or slowed down. In the peripheral nervous system, these myelin sheaths are formed from glial cells, known as Schwann cells. There are small gaps between Schwann cells, called the nodes of Ranvier. The action potential appears to leap from node to node, hastening the transmission. In the central nervous system, the brain, the sheaths are created by cells known as oligodendrocytes. Okay, so that's a quick overview of the neuron and how they communicate via action potentials. To recap, in the absence of a stimulus, the membrane is at its resting potential. A minor stimulus triggers a graded potential. A stimulus above the threshold initiates an action potential, causing the neuron to fire. So that's the potential journey of our neurons in action. It's polarizing, isn't it? It's positively amazing how this microscopic electrical dance forms the foundation of our interactions with the world. So charge forward with this newfound knowledge, and I'll see you in the next video.